Hello, animals, and welcome back to Biology 3013. Yes, I did call you an animal, and most of the time when somebody calls you an animal, they don't necessarily mean it in the most complimentary way. Maybe they're saying you're a little bit wild, you're a little bit crazy, or maybe they're saying you live like an animal and your room's a big mess. Well, when I call you an animal, I literally mean that we are organisms within the kingdom Animalia. So if you didn't guess it, today we are looking at chapter 17, the evolution of animals. We'll take a quick look at 17.1, but for the most part, we're going to focus our time on 17.2, looking through the different invertebrate phyla. So for 17.1, I said we are animals. What is an animal? There are many different characteristics that you have to be considered an animal. So first of all, remember, if we are thinking about the different classifications of life, we know that life is divided into 3 different domains. So we've got the bacteria and the archaea are 2 domains, and those different groups are made up of prokaryotic cells. And then we have the 3rd domain, eukarya, and animals fall within the eukarya. So that means that animals have to have eukaryotic cells, as do all organisms that are within that domain eukarya. Now remember, within the domain eukarya, we are going to have 3 major kingdoms: the animal kingdom, plants, fungi, and then we also have that catch-all group of protists that we have discussed already. So we're going to focus in within that eukarya, within that kingdom Animalia. So we've got the eukaryotic cells, and animals are also multicellular. So it's not one individual cell. Like we saw, many protists have one individual cell. Well, animals are multicellular. So they have lots of cells that are all going to work together in order to complete a common function. So they have to work together to be able to survive. Animals are heterotrophic. That literally means other feeders, meaning that they can't make their own food like plants. They can't do photosynthesis. They have to obtain their nutrients by others. And in particular, animals are doing this through ingestion. This is what breaks or separates them from fungi. Fungi, they release enzymes and break their food down, and then they absorb those little particles of food. Animals, they ingest their food first, and then they break it down inside of their bodies. Animals lack cell walls. We saw that many of the other eukaryotic organisms do have cell walls, those rigid structures around the cells. Animals don't have that. So as a result, they have to have something else to be able to help provide support to their bodies. So animals often have a skeleton. Maybe that's an endoskeleton made up of bones like you or I, or maybe it's an exoskeleton that's outside of their bodies that's providing them structural support. Most animals have muscle cells that allow them to be able to move, and nerve cells that are going to be able to interact with their environment, send signals throughout their body, and in particular, sending signals to those muscles to communicate to them when to move and in what way. All right, so there's some general characteristics of animals. There are most animals are also diploid. So remember, diploid means that they have their chromosomes lined up as homologous pairs. So 2 sets of chromosomes, one of which would come from mom, one of which would come from dad. There are exceptions, but most of the time the animals are going to reproduce sexually. So that's why they have the 2 sets of chromosomes, one from mom, one from dad. There's a lot of variation in how these organisms reproduce. Some of them are going to reproduce through internal fertilization like we do, but other animals reproduce externally, where the sperm and the eggs are released into the external environment and find a way to reproduce with one another. So because this is such a large group, there is a lot of diversity to it. So let's take a look then at how all of this diversity actually began. What was the origins of the animal kingdom? Well, we can't know for sure, but scientists hypothesize that animals evolved from a colonial protist. So we've got these protists, these unicellular organisms that started working together in a little colony. These were flagellated organisms, or so the hypothesis goes. And ultimately, these organisms, they were working together as a colony, and slowly over time, they would start to take on different structures to allow different cells to take on different functions. And then slowly over time, this colony eventually merged into becoming the first multicellular organism, where one of these protists, they wouldn't be able to survive without the colony. They all relied on each other so much that they couldn't survive individually. They could only survive together as a group. So again, that's the main hypothesis for how this happened. And this would have happened during the Precambrian period. And then it was during the Cambrian, and in particular the Cambrian explosion, where we started off with these simple organisms, and then we had a huge amount of diversity that just exploded all at the same time. Now, when we talk about the Cambrian explosion, we don't literally mean an explosion like something blowing up, but it's just referring to lots of diversity that's beginning to arise in a very, very short period of time. So this kind of explosion of life, explosion of different groups of life. So if we look at this diagram here, this image here, this is showing what the Precambrian seas may have looked like. So we're starting to see a lot of diversity here. And this is important because during the Cambrian explosion, the diversity that we see here is really the kind of starting materials for the evolution of animals. So all of the different groups of animals that we see today, they are the way they are because of different traits that they have that they originally got from organisms that lived in this period. If we look here, we can see a few examples of organisms. Up here, we can see this kind of fish-like organism is called Opabinia. And an interesting part is that it has these 5 eyes on top of its head that would allow it to be able to search for predators that were on top of it, keep an eye out to be able to escape those predators. Down here, this weird kind of stick-like bug is Hallucigenia, and it had these sticks for defense. So we can see how animals that have these mechanisms or these defense mechanisms, how they could have gotten these defense mechanisms from organisms like this that evolved during the Cambrian. There's no kind of consensus on how the Cambrian explosion or what caused the Cambrian explosion, but one idea is this idea of predator-prey relationships. And what that means is that there were predators that would eat these prey organisms, and then the prey organisms that survived were those that were a little bit better at being able to defend themselves from those predators. So maybe it had these stick-like structures that allowed them to be able to protect themselves. Maybe they were a little bit faster. Maybe they were a little bit larger. They had some kind of trait that allowed them to be able to survive getting eaten. Well, then those predators, slowly over time, they evolved to be more complex so that they could eat those prey that had evolved and become more complex. So this kind of back and forth of the prey becomes more complex, so it can't get eaten. Then the predators become more complex, so they can eat those prey. And then continuing. Eventually, over time, there was a lot of advancement within the diversity of animals because of these predator-prey relationships. Again, that's just one hypothesis that I'll mention on how there was such a huge diversification of animal life in such a short period of time. Let's see what else is on here. One important organism that I'd like to mention here are trilobites. So you can see trilobites down here. You can see a fossil of a trilobite over here. they were extremely abundant through the Cambrian, and they're actually extinct now, but they were these really neat organisms that had these exoskeletons that allowed them to be able to diversify and inhabit all kinds of different habitats. So there were over 50,000 different species of trilobites discovered through this time. And because they aren't around now, we could take a look at these trilobites through the fossils. And there's actually a lot of trilobite fossils because they had these hard exoskeletons that were great for fossilizing, but also because the sea floor at the time was very, very steep. So because it was so steep, mud would be able to kind of flow down the steep slopes of the seafloor and bury animals alive. And that allowed them to be able to, or that mud caused a great preservation of these animals so that they could later be studied through their fossils. All right, so we looked at a few examples here. There are more on there, but again, the main thing is that you can see that there's a lot of diversity that's starting here. All right, so then, what type of animals do we see today? What are the different groups of animals that we have? Here we can see a phylogenetic tree that is showing only 9 groups here. This is a shortened version of the tree. There's actually around 30 different groups of animals. The exact number of groups and the relationship between these groups, it's not actually fully agreed on by scientists, so it's always changing a little bit. But we're only going to look at these 9 major groups of animals. Now, if I ask you, what's your favorite animal? Think of your favorite animal. Maybe you're thinking like dog, cat, lion or tiger, elephant, monkey, horse, dolphin, shark. These are all kind of common examples of people's favorite animals. Well, all of those animals that I just listed, they all fall into this one little group here. So there's a lot of diversity of animals that we don't think about these organisms. Most of the animals we think about, they're in this one group called chordates, and this group we'll take a look at next week. And even within the chordates, you can zoom in on this branch of the tree to be able to see lots of other little branches. So most of the animals that I listed off there would actually just be on one small branch, in particular the mammals, within that category of chordates. So when we picture animals, we don't often think of all of the diversity of animals. So for this chapter, we're going to focus in on some of the invertebrate diversity. So these first 8 categories here are all invertebrates. And what that means is that they are animals without backbones. And they actually represent 95% of the animal kingdom. 95% of the animal kingdom don't have backbones. They're these often smaller, we often think of them as more primitive animals, but they are still animals and often neglected. So let's take a look at each of these first 8 different categories of the major invertebrate phyla. We'll take a look at some general characteristics about them, and then we'll take a look at some examples of organisms within those groups. Starting off with the most primitive group, the sponges. Sponges are stationary animals, meaning that they don't move, and they lack true tissues. They're also asymmetrical. They don't have any body symmetry yet. So these guys probably evolved very early on from that kind of general picture that we saw of the colonial protists. So remember, we saw that group of protist organisms, the flagellated protists that were working together in a little colony. Sponges look pretty similar to that, but then they have some extra cells that are more specialized to take on different functions. So if we look at the structure of a sponge, You can literally picture the body of a sponge being what we use as a sponge, or our kind of synthetic sponges that we use in the bath or to wash dishes or wash our cars, whatever. The sponges that we use today are synthetic, but the first sponges for cleaning were actually little pieces of this dead animal. So all the traits of, or all the characteristics of that sponge where it's full of holes, it's able to absorb a lot of water, well, that's the same traits that you see for the actual animal itself. So you can see here how it looks like a quite primitive animal. Scientists at first didn't actually know where to classify it. They said, well, is it an animal? Is it a plant? Maybe it's a plant. They thought maybe it was doing photosynthesis because they really couldn't figure out how this organism was getting food. But with further studies, they realized that it's an animal. And if you look over here, you can see how closely or how similar the structure is to the colonial protists that we saw on a couple of slides ago. Let's just go to this slide for a minute here. So you can see with that the protist colony here, we have these individual protists that are flagellated. And again, the hypothesis is that this would have been the first animals, and then eventually they started to rely so much on each other that they couldn't survive individually. Well, you can really see that structure when you're looking at the structure of a sponge here. So these cells here are flagellated cells similar to those protists, and the reason that they have a flagella is because they undulate that flagella back and forth, and that causes a water current to pass into the sponge and up out through the top of the sponge. And along with the water, there's going to be little bacteria, little phytoplankton, other little food particles that the sponge can then ingest. So on top of these cells here called choanocytes, on those choanocytes, there's a little mucus layer. So those food particles can get trapped in the mucus, and then the choanocyte can engulf those particles and then pass those particles on to a more specialized cell here. These little blue cells that look like amoebas, they aren't amoebas, but they are referred to as amoebocytes because they look similar to amoebas. So these specialized cells here, the choanocytes pass those food particles onto the amoebocytes, and then those amoebocytes will break down those food particles and pass the nutrients around to the other cells within that organism. So you can see that it's very similar to that colonial protist, except that it's going to have more specialization. Okay, let's move on to our next group, the cnidarians, or from the phylum Cnidaria. And organisms within this phylum, they have body tissues, they have radial symmetry. So remember, radial symmetry is like symmetry of a wheel or a pie. If you cut it through the center, you can cut it lots of different ways and still have two identical halves. And these are organisms that have tentacles with stinging cells. So can you take a guess at an example of an organism within the group Cnidaria based on those traits? Most people think of a jellyfish. So a jellyfish is one type of organism that you can find within the phylum Cnidaria. But there are two basic body plans that organisms can have within this group. So this polyp form is shown up here. This is a stationary or a sessile version, so it doesn't move around. It just attaches to a rock or another hard substrate and stays in place. You can see here that the cnidarians have a gastrovascular cavity. And what that means is that this is their digestive compartment, but it only has one hole into it. So that's the mouth, but it's also the anus. So it has double duty here. So the food has to pass through this opening into the gastrovascular cavity. The food gets broken down, and then it has to pass back up and out through that same hole. So you've got this two-way movement of food and only one opening for a gastrovascular cavity. So organisms within this polyp category, things like coral, sea anemones, hydras, even though they can't move, they're still carnivores. They still have to hunt for their food. So the way that they do that is they have these tentacles that are passing up into the water, and on those tentacles are their stinging cells. When a fish or another prey item passes by, they sting that prey, and then they try to wrap their tentacles around that prey and push that prey through their mouth, eventually digest it, and then the waste passes back up through that same opening. The second variation for the cnidarians are a medusa form. The medusa is this kind of flatter version with the mouth down, so it kind of looks like an upside down version. And this is a mobile version, so it floats around in the water and it drifts with the water, but it is also able to contract its body to help propel the water, or push the water to help propel this organism through the water. There's lots of different examples of jellyfish. I'll just name off the largest jellyfish is called the lion's mane jellyfish. And the umbrella, the top component of that jellyfish, is over 6 feet in diameter. The tentacles for a lion's mane jellyfish can be over 100 feet long. So very, very large jellyfish that we don't often want to think about. But even around here, we don't see lion's mane jellyfish around here, but we do actually have jellyfish in the Great Lakes. Most jellyfish are saltwater forms found in the oceans, but there are some freshwater forms too. So in the Great Lakes, we do have jellyfish, freshwater jellyfish, but they're very, very small. They're really just about the size of a penny, so they're rarely seen. They do have cnidocytes, so those are the stinging cells. But the cnidocytes for these jellyfish, because they're so small, they can't really penetrate into human skin, so they're not going to be able to harm humans. If you do get stung, if you're traveling, maybe you're out in the Maritimes or on the West Coast and you're in a more saltwater habitat and you do get stung by a jellyfish, well, don't pee on it. Many people say if you get stung by a jellyfish, then you should pee on it to try to take the sting away. Let me tell you, luckily not from personal experience, but it doesn't work. Not only is it gross, but on these tentacles, there are lots and lots and lots of those cnidocytes. So if you have a tentacle that gets onto your skin, some of those cnidocytes are going to fire, and that's what's going to sting you. But maybe not all of those cnidocytes actually fired. If you pee on that sting, well then that tentacle may still be there, and it's going to cause the rest of those cnidocytes to get released and actually cause you to get stung even more. So don't pee on it. The best thing that you should do is to just try to rinse your leg off or your arm off, wherever you got stung. Try to rinse it off in the water. Don't rinse it off with a water bottle, because the water in your water bottle is going to have different water chemistry as compared to the water that this organism is found in. So just rinse it off in the lake or in the ocean, wherever you are. Rinse that off to try to release that tentacle and try to ease the stinging a little bit. Okay, let's move on to our next group, the mollusks from the phylum Mollusca. These are soft-bodied animals that are usually protected by a hard shell, although there are some shell-less forms as well. Many mollusks feed with a radula that help them scrape up food, but there is a little bit of diversity with how they get food. Some of them are going to use the radula to kind of scrape up algae off of rocks, but some of them are going to use the radula to try to be able to break into prey organisms, to kind of chew apart prey organisms to be able to break them down and eat food that way. So a lot of variation. There's over 100,000 known species of mollusks, so a lot of variation. Most of them are marine, but there are some freshwater forms as well. If we look at the basic body plan for mollusks, you can see here, this is showing a general body plan, but it gives you an idea of the type of organisms that you can find within this group. So something like a snail is an example of a mollusk. And you can see here that mollusks have this muscular foot that allows them to be able to move. They have this visceral mass, which is just this fleshy component that's housing all of their little internal organs. And the third component is this mantle. And for the species that are able to produce a shell, it's the mantle that secretes that shell. So if we look at the different groups of mollusks, we're going to divide it into different categories. So again, there's so much diversity within the mollusks that we can kind of break that diversity down into three major groups. The first one are the gastropods, and this is where you find snails that do have a shell, but slugs as well, which are gastropods that don't have a shell. Gastropods, the shell that they have is typically a single shell that has this spiral structure to it. And then the snails, they're able to retreat and hide inside of that shell if they're feeling threatened. Now, an interesting part about snails is that you can see their body here is very, very soft. So when they hide into that shell, there's a big hole here. They have to be able to cover that hole to be able to be efficiently protected from a predator. So the way that they do that is they have what's called an operculum. And that operculum is basically acting like a trap door. So you can see here, this would be a snail shell. You can't see the hole underneath, but there would be a hole underneath here that allows the soft body of that snail to pass out through. If you look at this structure, this picture here, it's showing the same shell from the opposite direction. So this would be the hole that the snail is able to pass out through. When the snail wants to come out, it basically just opens this trap door so that the snail can come out. And when the snail wants to retreat back into its shell to be protected, then it's closing that trap door so that its soft body is completely surrounded by this hard shell and this hard operculum to be able to keep it protected. So a little neat adaptation of snails. Another neat adaptation of snails, of course, what many people think of when you think of snail adaptations, are these tentacles that hold their eyes. Because snails, their head here, it's so close to the ground, it's hard for them to be able to see. If you picture crawling around with your face on the floor, you're not going to be able to see very far. So snails have these tentacles that basically hold their eyeballs up so that they're able to see better distances. All right, the next group of mollusks are bivalves. Bivalves are mollusks that have two shells, and those two shells are hinged together. So they're able to open these shells, as shown here with this scallop. It's able to open to expose its soft body, or it's able to close those two shells tightly together in order to be protected. These organisms, they eat in a slightly different way than most mollusks. Rather than having a radula, the bivalves actually have what's called a siphon, which is basically just a little straw. So they can open their shells just a little bit and then pass this little straw structure out through their shells and basically suck water out from their surrounding environment inside. And then when that water comes inside, then they're breaking down the little bacteria, the little phytoplankton, little food particles that are in that water to be able to get their nutrition. The final category of mollusks are the cephalopods, which are amazing organisms. They are all marine and they are much more fast and agile. When you think of a snail, you don't necessarily think of speed. Cephalopods, though, even though they are closely related to snails, they are quite different. They are very quick. They are able to maneuver around very well. And they have these large brains and more sophisticated sensory organs that allow them to be able to detect where prey organisms are. And then they can swim over to be able to try to find that prey. Similarly, they also have the ability to be able to sense predators and try to be able to flee from those predators. Most cephalopods have a little ink pouch, and they're able, if there's a predator that's going to get them, they can release that ink, spray that ink at their predator that gives them just that extra little second to be able to try to flee away from that predator. Many cephalopods also have the ability to change their body structure and color to camouflage into lots of different environments. They have these color changing cells called chromatophores that are able to change color and change the texture of these organisms in the blink of an eye. It's really fascinating to be able to see how these organisms can change their appearance so quickly. So really just absolutely amazing organisms. If you want to learn more, I highly recommend the documentary by Netflix just from 2020 called My Octopus Teacher. And you can learn so much about octopi or octopuses from that documentary and see some of these things in action. You can see their ink pouch at work, and you can see their ability to be able to sense their environment if they're to be able to find food or if they're feeling threatened. So My Octopus Teacher, highly recommend checking it out. Our next category of animals are flatworms. So these are organisms in the phylum Platyhelminthes. And these are the simplest of all of the bilateral animals. So they have bilateral symmetry. They're ribbon-like and they have a huge diversity in their length. There are parasitic forms and there are free-living forms. If we go to this picture here, we can see a free-living form over here called a planaria. And with the planaria, you can see in pink its highly branched gastrovascular cavity. So it has the gastrovascular cavity that just has this one opening. So the planaria would be able to suck water and nutrients in through this opening. The nutrients in the water will pass through the gastrovascular cavity, and because there's so many branches, it adds more surface area to more efficiently be able to break down the organic matter that's inside of the water that it brought in. And then because it's a gastrovascular cavity, it's two-way movement of food, so those waste particles will pass back out through that same opening. On this picture here, you can also see some of the parasitic forms. So over here we can see blood flukes. Those are called schistosomes, and they are important, or they cause many important health problems, particularly in the tropics. They cause this kind of category of illnesses called schistosomiasis, and lots of different symptoms of this that is a result of these blood flukes that are going to attach on the inside of blood vessels in humans, and particularly near their intestines, so that they can get lots of nutrients. So if you want to learn more, then you can take a look at This Podcast Will Kill You. Check out their website, and they have a couple of episodes on schistosomiasis as well as other diseases caused by platyhelminthes. So you can take a look there. Another disgusting and yet fascinating flatworm is the tapeworm. And the tapeworm is a parasite that often has really interesting life cycles. So they often will inhabit different hosts. So they have intermediate hosts that they are in that intermediate host for a short period of time, just with the hopes of that intermediate host eventually helping this tapeworm get to its final destination, get to the inside of the intestines of the organism that it inhabits. So these tapeworms can parasitize lots of different vertebrates, but humans are one potential host for some species of tapeworms. And these tapeworms, they have these hooks and these suckers that allow them to be able to latch on to the side. Once they get inside of our intestines, they latch onto the side of those intestines, and then they can just absorb all of the nutrients, as many nutrients as they want, because there's so many nutrients in our intestines. It's a nice nutrient-rich location. So they can absorb those nutrients to be able to allow them to grow. As one example of a life cycle for a tapeworm, the tapeworm, its larval stage might be inside of a pig. And then when we eat that pork, if that pork is undercooked, then we can actually ingest that tapeworm larvae. And when we do that, well, then that larvae is going to end up inside of our intestines, and that's where it's going to be able to grow. That larval stage will be microscopic, so we won't actually know that we're eating this little tapeworm larva. But then once it's inside of our intestines in that nutrient-rich location, it can grow quickly. And it can eventually cause issues, because if it grows large enough, it can block our intestines so that food can't pass through. But it can also cause nutritional deficiencies if it gets large enough that it's stealing enough of our nutrients before we are actually able to digest them and absorb them ourselves. So tapeworms, if you do get a tapeworm, you can have an orally administered drug that can kill the tapeworm. And if it's small enough, then you're just going to poop it out. But if it's a larger tapeworm, you might need to have surgical intervention as well. The longest tapeworm ever found in a human, so picture a human's intestines, the tapeworm that was the longest tapeworm ever found inside of a human's intestines was 82 feet long, 25 meters long. So you can imagine how that would block that human's intestines and cause nutritional deficiencies as well. Okay, so let's move on to our next group, even though I hate moving on, because although these guys are completely disgusting, they're such a cool organism, and there's so many fun characteristics about them. But let's move on to the next group, called the annelids, within the phylum Annelida. These worms have body segmentation, so they're made up of these repeating units. And you can best visualize that body segmentation when you're picturing an earthworm. So an earthworm is just one example of an annelid, and you can see within its body plan here these repeating units. You can also see in this diagram that the annelids have a complete digestive tract. So you've got a mouth here, then you've got this kind of light pink tube would be its digestive tract, that it's going to pass through that entire organism, and on the other end of the organism is where the anus is going to be. So you have one-way movement of food from the mouth through the organism out through the anus. So that's in contrast to the gastrovascular cavity, where there's two-way movement of food and you only have one opening, the mouth/anus. So here, rather than a gastrovascular cavity, you have a complete digestive tract. If you haven't figured it out yet today, the kind of theme of the day is diversity. All of these groups have so much diversity, and the annelids are no exception. There's lots of different species, and the annelids are divided up into three major categories. The earthworms, which are going to eat through soil and they extract the nutrients from the organic matter within the soil. The polychaetes, which are a marine form. And then leeches, which get a bad rap here. When we think of leeches, we think of the blood-sucking leeches. There are lots of different types of leeches. Most of them are actually free-living carnivores, but some of them are parasitic. Some of them are the blood-sucking form that you can end up getting on you when you're going out for a swim in a lake. Moving on to our next category, the roundworms, our last category of worms here. There have been a lot of different worms within the animal kingdom. So the roundworms are found within the phylum Nematoda. They're cylindrical in shape and they're tapered at the ends. And what that means is that they come to kind of a point rather than being a more rounded off end. And these guys are, again, extremely numerous and extremely diverse. There's lots of different parasitic forms, but many free living organisms as well. There's actually a huge number, so not only diversity, not only different species, but a huge abundance, a number of organisms of these free living roundworms that are important decomposers. I actually read at one point that one single apple that fell off of a tree and is rotting on the ground was found to have over 100,000 of these little roundworms in it, chewing up that apple and breaking it down, so helping to decompose that apple. The next group that we're going to look at is the most abundant and diverse even yet. Organisms from the phylum Arthropoda, and they're named arthropods because of their jointed appendages. So arthro means jointed, pods means legs, so they have jointed legs, but it's not just their legs, it's actually legs, claws, feeding appendages, mouth parts. They have lots of different little appendages coming off of them, and they are jointed, meaning that they're able to bend, so they're able to take on lots of different functions. The arthropods are the most successful animal phylum on Earth, and they are so abundant that scientists can't actually agree on how many arthropods there are on Earth. Estimates range, but one estimate is that there are approximately 150 million arthropods for every one individual human on Earth. So there's one of me, for every one of me, 150 million of these guys. That is a lot of organisms. So arthropods can be broken down into 4 major categories. We'll take a look at each one of these categories and talk about a few characteristics and some examples. So the first category of arthropods are arachnids. Sorry for those who are out there who are arachnophobic. It is a legitimate phobia. I understand that. So if you have to flip to the next slide, please do so. Arachnids, they primarily live on land. The organisms within the arachnids that we are most familiar with, of course, are the spiders. So we know a lot about spiders from our childhood, catching spiders, looking at webs, or maybe that's just my childhood. But as I say that, I remember as a child, we would take a piece of paper and put it behind a spider web and then use some spray paint on that spider web to be able to catch the outline of the spider web on our paper. So a really neat little project for anybody out there who has any kids who likes to go out and try to explore nature. So arachnids, they have 4 pairs of walking legs, and then they also have a specialized pair of feeding appendages as well. Even though we think of spiders when we think of arachnids, there's actually a lot of diversity. Scorpions are found within the arachnids. There's poisonous and non-poisonous forms of scorpions, and it's their tail part here that does the stinging. So when you see their tail raised up like this, that's when they're feeling threatened, and they have that stinger ready just in case they're going to need it. So if you happen to encounter a scorpion and you see its tail up like that, then get out of there. If its tail is down, then it doesn't feel threatened. But once that tail goes up is when you have to get concerned, particularly if you're in a location where there could be poisonous scorpions. Other species within the arachnids, there are mites like dust mites, and then there are ticks, which we've talked about the black-legged tick as one example that is able to transmit Lyme disease. So these are all examples of arachnids. The next category within the arthropods are the crustaceans. Crustaceans are nearly all aquatic and they have lots of different appendages to them. They have walking legs, but they also have these different feeding appendages. They have these claws that they can use for defense or to be able to try to capture prey. You can see here, these are feeding appendages called cirri that are on, these are barnacles. So lots of different appendages. And again, they can take on different structures and take on different functions because they are jointed. Now, as we look here at the barnacles and we look at the cirri or these feeding appendages, I just want to talk about barnacles just for a minute because they are absolutely fascinating. And the reason I say that is because barnacles, they have plasticity in their cirri size. And what that means is that plasticity is that the length, the dimensions of those feeding appendages, of those cirri, they're actually able to change based on the water environment that they're in. So I drew out a little diagram here to help. First, this picture here is showing the cross section of a barnacle, and you can see the cirri shown here. So the way that the barnacle feeds is that it's going to open up its top plates here, and it's going to release its cirri out into the water, and then the water would flow through these cirri and be able to filter out any food particles that are passing through the water. Now, the barnacles that live in different locations are going to have different sizes of these cirri. In a low water current environment, these cirri are going to be much longer and thinner as opposed to cirri that are in a high water current environment. In a high water current, the water pressure is going to be pushing on those cirri so much that if they were long and thin, then they aren't going to be able to stick up out into the water. They're just going to get pushed around, so they're not going to be effective at being able to filter out food. So in a low water current, you've got these long, thin cirri. In a high water current, you've got these shorter, stockier cirri. Now, the plasticity part is saying that if we took barnacles that are naturally in a high water environment, so they naturally have these shorter cirri, if we took those barnacles, maybe they're on a little rock, and we take that rock and we put it in a different environment. We put it in a low water current environment. Then slowly over time, you're going to see those shorter, stockier cirri. They're actually going to grow longer and thinner. That's going to help them be able to feed better in that low water current environment. The opposite is also true. If you take barnacles from these low water current environments that have these longer, thin cirri, and you transplant them into this high water current environment, those cirri are actually going to grow shorter and stockier to allow them to better feed in that high water current environment. Now, one last thing with barnacles. I can't move on without talking about the barnacle penis size. Yes, you did hear me right. We are going to talk about barnacle penises, because they are also fascinating. Don't judge until you understand why. So, we said that the cirri are long and thin in these low water current environments and shorter and stockier in these high water current environments. The same is actually true for their penis size. Now, the reason is barnacles, even though they're sessile, so they're stationary, they are stuck on these rocks or on these other hard substrates, and they don't move. They spend their life there. So they don't move, and yet they have internal fertilization. So what that means is that the penis actually has to pass out through one barnacle like here, and then go and fertilize the inside of the other barnacles nearby. So the penis actually has to enter inside of another barnacle in order to fertilize it. So you can imagine that having the longer penis size, if we just take this here and let's just we'll say that this penis belongs to this barnacle here. You can imagine by having that longer penis size, well, it's going to be able to then fertilize lots of barnacles that are around it. So having the longer penis size is going to be an advantage when you think of reproduction. Now, if we grab this shorter, stockier penis size over here, and let's put it on the same organism, this is a high water current environment. So this water is really pushing in this direction. So the penis, it has to be able to withstand that water pressure. So it's going to be able to fertilize over here and over here. It's going to be able to fertilize over here. And then because it's shorter and stockier, it can push against the water current to be able to fertilize on the opposite side a little bit. If you picture having this longer, thinner penis in this high water environment, well, then it's just going to get pushed around by the water current. It's not going to be able to fertilize in this direction over here because the water is going to push it away. So having that longer, thinner penis is actually very limiting in that high water current environment because it's going to get pushed around by the water. So again, it would be an advantage to have this longer, thinner penis in a low water current environment and this shorter, stockier penis in this high water environment. The plasticity part still applies here. So if you take a barnacle from over here that has this shorter, stockier penis and you put it into a low water current environment, then its penis size is actually going to change over time to become longer and thinner, similar to the way that the cirri change. So it's a really, really neat aspect of barnacles and the plasticity that barnacles have. Little tidbit for those who are interested. Not sure if anybody's heard of the expression hung like a barnacle, but it's legit. Barnacles actually have the largest penis to body size ratio of the entire animal kingdom. So that expression, let's get it going. Hung like a barnacle. All right, let's move on to our next category of organisms within the arthropods, and these are the millipedes and centipedes. Millipedes, as shown here, they have two pairs of legs for every body segment, whereas centipedes just have one pair of legs for each of their body segments. So they don't actually have 1,000,000 legs, or they don't actually have 100 legs, but they have two pairs of legs for every body segment for millipedes and one pair of legs for every body segment for centipedes. Even though millipedes may look a little bit scary, they actually aren't. They aren't harmful. They are decomposers, or they're breaking down material on the forest floor. Centipedes can, however, be dangerous. There are poisonous versions of centipedes, but the version you're probably most familiar with is the house centipede, shown here. House centipedes often live in basements of houses. They like damp environments. So maybe if you're down in your basement, or if you live in a basement apartment, maybe you've seen some of these guys around. We have a tendency to freak out and get scared, but these guys are not harmful to us. They're actually insectivores. So maybe you want to keep them around your house, because all they're going to do is they're going to eat other insects and spiders, and they're not going to harm you. So it is a type of centipede that's called the house centipede. The next category of arthropods are insects. Insects, which are the most successful group of organisms in the entire animal kingdom. They have a 3-part body plan where they have a head, thorax, and abdomen. They have lots of little appendages coming off of them. So they often have 3 pairs of legs. They often have wings. They have various sensory organs coming off of them, like little antennae. So lots of different components to them and lots of variation from one species to another. The main reason why insects are likely so successful is because of the adaptation of wings. Because they've been able to fly, it allows them to be able to inhabit different locations and allows them to be able to more efficiently capture prey, allows them to be able to more efficiently escape predators. So the evolution of wings was key for the success of the insect group. You can see here just a few examples of insect diversity. There's so much that you can take a look at with insects, so we can't go through it all, but you can see here a couple of examples of different types of beetles. So a rhinoceros beetle here, a weevil shown here. They have these hard outside wings that they can just kind of pick up those plates, and underneath that hard plate they're going to have their softer membranous wings underneath. So these hard external plates can kind of protect those soft membranous wings, and then when they want to fly they can just lift those plates, releasing their membranous wings to allow them to be able to fly. You can see here different wings are going to take on different structures. The butterfly wings are much larger and broader as compared to the dragonfly wings that are going to be much thinner. So there's a huge amount of diversity within the insects. Insects go through a process called metamorphosis, which I'm sure we're all familiar with. And the classic example of it is with a butterfly. So most insects go through complete metamorphosis. And what that means is that their larval form is very different than the adult form. So the larval form looks differently, and the advantage of that is that it often lives in different environments, feeds on different food, and as a result, the larvae and the adults aren't competing with one another for the same resources. So if we look at this example of metamorphosis here, we know that the larval form is the caterpillar for the butterfly. And then that caterpillar is going to go through a series of molts before it eventually emerges as its adult form and is able to fly off. Now, this is the example that most of us would be the most familiar with, but it's not just butterflies that are doing this. All insects are doing this, but it's not always as visible as something like a cocoon is. So many insects actually have their larval form as an aquatic form, meaning that they lay their eggs in the water, their eggs develop into the larva in the water, and then go through their series of molts underwater until eventually they emerge from the water as the terrestrial flying adult form. Some insects, they actually spend years underwater as this larval form developing, and then just minutes, hours, or days as the terrestrial form. And the primary function of that terrestrial form would simply be to go around to mate, to lay its eggs, and then ultimately die. And then those eggs would then restart the cycle, develop into the larvae, go through this series of molting, and then ultimately hatch as an adult again. Now, because of the different series of molting that these insects go through within metamorphosis, it allows for a neat field of science called forensic entomology. So an entomologist is a scientist who studies insects. A forensic entomologist is an entomologist who in particular is going to use their knowledge of insect life cycles to help solve criminal cases. So I'm sure we've all seen a little bit of forensic entomology on TV before, on different crime dramas, maybe like CSI, where the forensic entomologist is going to look at the maggots or look at the larval form of that fly, oftentimes, maybe a blowfly, and that forensic entomologist is going to analyze the size and the traits of that particular larvae to determine how long it has been on that corpse, and it gives you a better idea of time of death for that particular organism. All right, the last group that we are looking at are the echinoderms. So we're out of the arthropods, we're back just into animals, and this is the last phylum that we're looking at for today within the animals. So this is the phylum Echinodermata, and the organisms within this phylum typically show radial symmetry as adults, but they actually often have bilateral symmetry in their larval form. These organisms have an endoskeleton, so they have structural support that's beneath their skin. They have a water vascular system that's this network of canals that fills with water, and it circulates water around through their body, and it helps for that organism to be able to move, but it also helps with gas exchange and waste disposal as well. So if we're trying to think of examples of echinoderms, have you figured out what they are yet? Echinoderms are things like starfish, or also called sea stars. Sea stars, where if you zoom in here, you can see they have these little tube feet. These little tube feet are part of their water vascular system. So these tube feet just fill with water, and which tube feet fill with water and when is what's going to control the movement of that organism. You can see here with this sea star here, it's wrapping its legs around this clam. I think that might be a clam, can't tell for sure, but around the clam, and it's going to try to form a small crack in the clam, in that clam, to then be able to feed on it. So sea stars, they are carnivorous, as with most echinoderms. You can see up here a sea urchin, and you can see it's kind of an upside down form. All of its tube feet are up here instead. The sea urchins, they aren't carnivores. They typically just graze on seaweeds, algae, whatever they can find around. Here we can see a sea cucumber, which is really neat because they have these 5 rows of their tube feet. These guys are often scavengers, so they're going to feed on dead material that often drops down to the sea floor. The last example here is a sand dollar. A sand dollar, which is a really neat organism that I remember finding sand dollars as a kid when I was at my family cottage on the coast of Nova Scotia. And what I eventually learned is that what I was finding wasn't an alive sand dollar, it was the endoskeleton of that sand dollar. So sand dollars, remember with echinoderms, they have that endoskeleton. They have the shell, or it's kind of like a shell that's on the inside, and then this skin that's on the outside. Well, the sand dollars that I would find as a kid, they were all white. And so I would pick them up off the sand, and I'd throw them back in the water thinking I was saving them, except that the all white ones are actually just the endoskeleton of that sand dollar. So it was already long dead. But sand dollars are really neat. They have little tiny tube feet on them that allow them to be able to move around on the sea floor. And again, they're going to, similar to the sea urchins, they're going to eat things like algae. They can eat some other animals like little zooplankton, little kind of unicellular animals that are around, but they're not going to be killing large things like fish to be able to eat them. So the sea stars, sea urchins, sea cucumbers, sand dollars, all examples of echinoderms. And remember, echinoderms is our last group of invertebrate phyla when we're looking at the animal kingdom. So that's where we're going to stop for today. Chapter 17 is a long chapter. So next week we will pick up from here and we will start to take a look at chordates and some of the diversity within chordates. As always, if there's something from the chapter that you don't understand, please reach out to me by email and I'm happy to help you out. And if not, happy studying!